Flowchart illustrates the effect of a point mutation. Abinine, missense. Do you mind clarifying what mutation that was? Because I asked you that last lesson, so I want to get the recap answer. What is a missense mutation?
So when you get the codon changing, I guess, due to the base substitution, or it's a stop codon, whatever.
0 out of 1 so far. Maya, your turn. What is a missense mutation?
A change in a substitution in a single base that results in a change in one amino acid.
Good, good. So the key thing here is when I ask you all questions in this lesson, you need to give me exam answers back. I'm going to quite literally mark you on the spot based off your verbal answer, because if you can't verbally say it, you can't write it in an exam. There's no hope there, right? So when you think about a mutation, you should all by now be able to say to me that it could be at a chromosomal level or at a base level. And a base level is a point mutation, right? And with a point mutation, you can have a substitution, or you can have a frame shift, which is your addition or deletion. If you have a substitution mutation, Micah, what are the 3 types you can have again?
Missense, nonsense, or silent.
Good. And missense, what's the key buzzword you want to have in a definition?
Like it's a change in one amino acid.
Very good. That's what I was looking for, Abhinay, in your answer. It's a point mutation, a point substitution mutation that results in a change of one amino acid. Not stop codon, because what is a stop codon mutation, Miriam?
A stop codon mutation is a nonsense mutation.
Yeah, very good. So nonsense mutation produces a stop codon. Good. I mean, do you just mind muting yourself? Only because there's a background frequency that I can hear. Yeah, thank you. You feel free to unmute yourself, everyone, when I do ask you a question, though. Good. So nonsense mutation produces a stop codon, and silent, produces the same amino acid. Good. Now, what you see here is a point mutation, and what has occurred? You've gone from TCT to where? ACT. Do you agree? That's the effect here. It's a base substitution. What's happened here? What is this representing? Number 3, Sean?
Is that meant to be the RNA to it?
mRNA.
mRNA, sorry.
Very specific. Okay, remember, 2 weeks out from the HSC, you have to be very specific. Bio is not hard. It's just an exam of articulation and specificity. So focus on the detail there. Good, that is the mRNA strain. You see uracil, so straight away you think mRNA. So what do you think you should be representing here? Because I stressed this to you. With a long response about mutations, you need to link 3 things. What are the 3 things you need to link? If they say this mutation has happened, how's it going to affect the organism? What are the 3 things you talk about?
How it affects the base sequence, and then the polypeptide, and then the protein.
Yeah, so the way you should be thinking of it is, how does it change the DNA? So what kind of mutation is it? So identify that, right? And then you have to explain the effect on the RNA, then the effect on the amino acid sequence, and then you link it to the polypeptide and the phenotype. Does that make sense? So that's the structure I have. Make sure you engrail that into you remember that, okay? Because it's very important. Good. All right, so outline the series of events from stages 2 to 4 that resulted in the faulty protein. So here we know we are talking about the amino acid sequence, right? What did I tell you? Look at the DNA, then you must look at the RNA, then you must look at the amino acid sequence. So series of events straight away. Let's see. Ishan, what are they actually testing you here?
Like what they're questioning us on.
I'm looking for 2 words. You either know it or you don't know it. What are the 2 things?
Transcription, translation.
Very good, right? That's all you need to realize. So the question is quite literally outline transcription and translation, but refer to the letters you see up here, right? So Ishan, now's your chance to explain to us, the class. Can you describe transcription to us? Exam answer. I don't want to, ifs and buts or some things and things. What happens in transcription? Clear, concise answer. Go.
In transcription, mRNA mRNA template is formed, which can I don't know how to word it.
Okay, let me ask Braden.
Can I go?
Yeah, Zachariah, give it a go. Sorry, I
Right.
couldn't see your picture, so I didn't ask you too much. Good.
That's all right. The process of the purpose of transcription is to create a template strand of DNA that is able to float into the cytoplasm and reach the organelle. Damn it, how did I forget this? The ribosome, where it is translated in the next stage. For transcription, the template is created via damn it.
That's okay. That's okay. This is why I'm putting it out. So you all realize that you all know this stuff, but you all need to work on your articulation. Right now, your articulation for all of you is not at a full band 6 range yet. So you need to focus on fine tuning that. Maya, your turn. I want one sentence and I want the keywords. Go.
In transcription, mRNA is a complementary mRNA strand is created from the DNA template using RNA polymerase.
Perfect. She's mentioned complementary. She's mentioned RNA polymerase. And it's the only other thing I'd mention is that uracil is replaced instead of thymine. If you mention these three things, right, you've got the crux of transcription. Remember, this is a 3-mark question. If it was 5 marks, for example, then you start talking about 5' guanosine cap and polyadenine tail, poly A tail, all of that stuff, and splicing out your introns. All of that is extra. You don't need it. And this is why you need to do exam questions, right? Students know all this stuff, but the poor students will just verbal garbage everything they remember. Good student would know what are the key things I'm looking for in an exam. If you don't say the word complementary, you're not getting the mark. If you don't refer to the letters here, you are not getting the mark, right? So a template exam answer would look like this, right? In step 2 to step 3, the process of transcription occurs, whereby the template GATACT strand is converted into a complementary mRNA molecule with thymine replaced by uracil. The enzyme responsible for this is RNA polymerase, full stop. You see how I referred to the data? I've used the buzzwords, and it's clear and concise. Everyone with me, right? Now, I don't want to waste too much time. You guys should be a bit faster with this. I'm going to pick one person to do better and explain translation well. So how about, Miriam, throwing it to you now. Translation, what would you like to mention?
The mRNA strand travels to the ribosome, which and then it is formed into amino acids, which bind into groups of 3, creating codons, which then bind, creating a polypeptide.
Good. But why did you go from amino acids back to codons? Because codons are still the language of mRNA. And the second you say amino acid, the mRNA is coded for the amino acid. You don't need to mention codons. The mRNA is structured as codons. Does that make sense? So being specific here, you need to mention mRNA codons will affect the tRNA anticodons and the amino acid sequence. And one thing you missed is a tRNA that I'd like you to mention, right? One more person. Let me ask Raiden. Give it a go. Translation.
In translation, so mRNA travels to the ribosome where it's decoded outside the nucleus and it produces a amino acid chain.
Okay. Now, with this question, is there anything you'd like to mention, Raiden? Specifically, because you gave a generic description that could answer any.
You have to mention the mRNA to the amino acid from step 3 to 4.
Yeah, Good. Any other data we'd like to mention, everyone, to be safe? To show that we're answering this question and not a general.
CUH, UGA.
Either 3' to 5' direction.
No. That's going way outside this question. There is no need to mention 3' or 5'. Do not mention that unless it is given in the question. Okay, that's a big learning point. Very low yield. Come on, guys. How many codons do you see here? You mentioned the word codon in all of your answers, but why is no one
Just
talking about
two.
the number of codons? Yeah, so how many amino acids would this produce?
2 amino acids.
Good. And why would the protein be faulty, everyone? What does it mean to be a faulty protein from a substitution mutation? 2 mutations could have happened to make it faulty. What are the 2 mutations? Abinan, your turn. What 2 mutations from substitution can turn the protein faulty? I gave you 3 here. Kind of
Is it
literally
missense and missense and the one which changes the stop codon?
Yeah,
Stop
I don't
Nonsense.
yes,
Yes.
nonsense. Very
Yeah.
good. I don't want to hear this one or that one. I want specific answers. So missense or nonsense mutation, right? So to be very
Prithvi,
specific, yes?
I was gonna ask, with the nonsense mutation, since it's non-functional, would that be a faulty protein, or is it not counted?
A nonsense would be a very faulty protein because I guess the way you think about this is not the entire amino acid sequence, the GATTCT, right? It's much longer than that. So imagine there's another 500 amino acids in the normal polypeptide. If you cause a nonsense mutation and you produce a stop codon, which quite literally just codes for release factor. So instead of an amino acid binding, release factor will bind, and everything to the left of that is going to get released, aka that's going to be the final polypeptide. You're not going to produce whatever 500, if this was 500 bases, whatever 500 divided by 3 is, you're not going to produce those many amino acids, right? So you're going to have a severely deficient in size polypeptide chain. Does that make sense? Nonsense is the
Yep.
worst. It's going to make a worse faulty protein than a missense mutation, right? So what I would mention, the key things I'd mention here is translation is a process whereby the ribosome will move along the mRNA codons, attaching complementary tRNA anticodons in a specific sequence of amino acids. Full stop. Do you see how I've spoken about codons, tRNA, amino acids?
Yep.
Yes?
I was
That's it.
just a bit confused because our teacher told us that if a mutation is generally deleterious, then you could say the protein is faulty. Otherwise, it's not. And I wasn't sure with like nonsense
The
because
mutation is
the protein
deleterious.
isn't even made.
Deleterious just means bad in broad terms. It's just a fancy way of saying bad, right? And so it can be bad through a nonsense mutation or bad through a missense. So both ways can give you
Okay, fair enough.
a faulty protein. Yeah, good. So what I'm looking for all of you saying here is basically the keywords I mentioned: ribosome, mRNA codon, tRNA anticodon. Sorry, Abinay, just do you mind muting yourself? Only because of the friction. Yeah, thanks. Good. So ribosome, mRNA codon, tRNA anticodon, and I want to also hear you mention that as a faulty protein was produced, this likely would have resulted in a missense or a nonsense mutation. Right? Good. And you can maybe be specific and mention 2 amino acids were affected here. So if you can put that into 2 sentences, you have all the buzzwords there. Okay. Good. Do you see how we're answering the question but also being specific to the question? I think that's what you're all missing, that little fine tuning where you need to answer the question and not just generic description of a process. Okay. Good. Micah, every time they give you data, what are you going to do to that data in the question?
Mention it.
Good. Not just mention, but try to put it in some kind of calculation. For example, if they give you 50 bases, you know it's 50 divided by 3 amino acids. So state the number of amino acids. That shows you understand what a codon is, right? Good. Describe the effect caused by mutation and the effect what this would have on the organism. Okay. So we kind of already spoke about that. The effect here is that it would have resulted in either a missense or a nonsense mutation. as a silent mutation would still produce the same amino acid sequence and still produce a functional protein. So what effect could this have in the organism, everyone? How would you explain this? I'm going to pick Maya. You have 2 lines to explain how these mutations would affect an organism. You can use 3 if you'd like, but give it a go.
A missense or nonsense mutation would result in a faulty protein being produced, which affects phenotype. Yeah, I'm not really sure.
Maya, you wasted 1 sentence because you repeated the question there. The effect it would have on the organism, right? And it says here it's a faulty protein. So you just repeated mutation, faulty protein, phenotype. So we're back at square one here. So Miriam, your turn. What would you say?
I would say the faulty protein could end up coding for a different process than what it was meant to do, or if it was a nonsense mutation, it could end up with a dysfunctional protein due to the premature stop codon.
You know, both would result in a dysfunctional protein. One would just be more dysfunctional, but let's not focus on that. Now, again, I'm going to repeat the same question I've been asking you every single question, because I think...
Can I have a go? I'm not sure this is right, but...
If you answer my question first, Bilal, which is what
Okay.
is this question asking you? Really? What is it really asking?
The effect it would have on the organism?
You guys are just repeating the question to me. I have eyes I can read. You know, I have a functioning retina. Come on, guys. This question is testing your understanding of protein structure and function, right? So the question's told you you make a faulty protein. They don't want to keep on hearing, you know, the protein is faulty, the protein is dysfunctional, the protein is denatured. We've heard that enough times here. You need to explain how that's going to affect phenotype. Well, what do proteins do, Micah? What were the 2 categories we had?
Structural proteins.
Good, and?
Enzymes.
Yeah, functional. It doesn't just have to be enzymes, right? You're all thinking right now, do you know what neurotransmitter you're using to think, relay information, strengthen connections between brain cells? Right now, your brain, Maya, do you remember watching Harry Potter and the staircases that keep on moving?
Yeah.
The library? That's like your brain right now, especially at your age. Your neurons are wiring and firing and forming new connections and breaking old ones. Your brain is growing and changing, right? The reason I'm bringing this up is the neurotransmitter that allows you to do this is glutamate, which is a protein. Proteins have very broad roles. It's not just enzymes. It's not just enzymes for functional. They can act as neurotransmitters. There are protein hormones. You remember insulin, everyone? Insulin is a functional protein, right? It's an Uber, it's a microscopic Uber that takes glucose and drops it off into a cell. Good. So it's broader than that. So I want to just hear structural and functional. So you need to explain that the structural protein could be impaired, right? Or a functional protein could be impaired if the protein was a functional one. And you can provide examples there. All right? And then you'd be answering the question, right? And you can talk about missense and nonsense as well. So you can mention a missense mutation may have occurred where one amino acid was altered in this protein, rendering it dysfunctional, or a nonsense mutation could have occurred whereby a premature stop codon codes for release factor and produces a prematurely terminated polypeptide chain. Then you need to explain the dysfunctional protein will affect phenotype as it can either serve a structural role or a functional role. Its structural role could be impaired, e.g., you can mention a structural protein actin myosin in muscle fibers, causing disease. If this protein were to serve a functional role, it would result in a biochemical change in the organism, e.g., inability to break down a compound, which can also result in disease. Give an example. Do we know any really famous examples of an enzyme and a disease it causes? Very famous one. It was brought up in your 2019 trials and it was on all the HC discussion groups and students were complaining that I didn't understand this.
Phenylketonuria?
Very good. Phenylketonuria, PKU. PKU or phenylketonuria. And Maya, do you remember what that reaction involves?
Not really.
So there is an amino acid tyrosine, and it's usually found in normal Coke. It's found in meats. It's found, I believe, in some milk, and it gets broken down into phenylalanine. And the way it gets broken down is OH groups gets taken off it. So we call the enzyme a hydroxylase. Those of you who do chem would know OH is a hydroxyl group. The enzyme here is called phenylalanine hydroxylase. Right? And you can have an autosomal recessive disease whereby you can't produce this enzyme because of a mutation, just like in this case here. And so what happens is you cannot eat tyrosine because the tyrosine you eat cannot get broken down anymore into phenylalanine. If it builds up in the blood and you eat tyrosine, you can cause damage to nerve cells. So you can cause mental retardation, seizures. And you typically see this in blonde Caucasian kids. And if there were a patient in front of you, if you're a doctor, one of the symptoms they might have, you might notice a musty odor. It's quite weird. That's what tyrosine produces when it builds up in the body. Yeah. Good.